Hey everyone, and welcome to this mark scheme specific and examiner's tips specific video all about neuromuscular junctions and muscle contraction for topic 6 for AQA. So this video is going through everything you need to know, but really focusing on common exam questions, the mark scheme and examiner's feedback as well. So let's start by looking at the neuromuscular junction. Striated and skeletal muscle contracts when it receives an impulse from a motor neuron. So this is the connecting points between what you would have learned in the nervous system to then in muscles, because muscles are an effector. So through the neuromuscular junction, we then get the neurotransmitter diffusing across, which is very similar to a synapse. And this is then what results in the muscle contracting. But we're going to go through exactly what is happening in mark scheme specific detail. So you can write these down as notes, or you can turn it into flashcards, whatever you want. So let's go through it step by step. Number one, we have an action potential will reach the presynaptic membrane of this motor neuron. Now on each bullet point, I'm highlighting the key terms that you would have to have in your answer, which are often underlined in the mark scheme, meaning you have to say them to get the mark. Step 2 then is this causes calcium ion channels to open, which are within that presynaptic membrane, and therefore the calcium ions will diffuse into the presynaptic neuron. So again, we've got highlighted the key things you'd need to make sure you definitely say, and diffuse is always a key one. And also really, you should be mentioning that it is the presynaptic neuron as well. That influx of calcium ions will cause vesicles that contain the neurotransmitter, which might be acetylcholine, to fuse. So they actually move towards and then fuse with that presynaptic membrane. And that causes the release of the neurotransmitter, such as acetylcholine, across or into that neuromuscular junction. Now, a few things to point out here. I have abbreviated acetylcholine at this point, but do not do that in the exam unless they provide it to you in that way in the question. Or as I've done here, I've written it out once and then I've abbreviated it to show that I know it's this molecule and now I have abbreviated it. Another thing to point out is you would have to say that it fuses with the presynaptic membrane as well. Our next mark then is that neurotransmitter or the acetylcholine diffuses across that neuromuscular junction, and diffuse across is always a key marking point. And we can see at this point it's quite similar to the description of a synapse, so that would be another thing with a synapse you'd have to say diffuse across as well. Once it's diffused across, it then binds to receptor proteins on the sarcolemma. Now this bit is really specific, so you do have to say it binds to receptors, and then the place that it binds to is the receptors on the sarcolemma, which is basically the muscle fiber's surface membrane, which we can see here. But that's one part that students often don't say or they get wrong. So really make sure you're saying that it's binding to receptors that are found on the sarcolemma. That binding then of the neurotransmitter to the receptor causes sodium ion channels within that sarcolemma to open, and that then enables the sodium ions to diffuse in. So again, on this one, we can see underlined is ions. You'd have to really emphasize it's sodium ion channels that open and then sodium ions diffuse and enter. Now, once those sodium ions have entered, the process carries on. Those sodium ions diffusing into that sarcolemma is going to cause the membrane to depolarize, which means it's starting to become more positive, and that's because the sodium ions are diffusing in. And if that reaches that minus 55 millivolt threshold, then an action potential will be generated. And that action potential then moves down the T tubules towards the center of the muscle fiber. Now the T tubules bit is often not on the mark scheme. That's a bit of extra information, but if you're writing an essay, it'd be really good to have that in it. So we can see here, this is showing you that surface of the muscle fiber, and it moved down the T tubules so it then reaches the center. Those action potentials will prompt the opening of voltage-gated calcium ion channels in the membranes of the sarcoplasmic reticulum situated near the T tubules. So that's what we can see here. We've got the sarcoplasmic reticulum, which is near those T tubules, and calcium ion channels are opening on those. As a result, the calcium ions diffuse out of the sarcoplasmic reticulum and into the sarcoplasm, which surrounds the myofibrils. So that then enables the contraction of the muscles via the sliding filament theory. So just to summarise some key similarities, well, there's actually only one, the rest are differences between the neuromuscular junction and a cholinergic synapse, which means it's a synapse with the neurotransmitter acetylcholine. Both of them involve the unidirectional or one-directional movement of the neurotransmitter, and that is because you only have the neurotransmitter receptors on the postsynaptic membrane. A neuromuscular junction is always excitatory, meaning it will always result in the generation of the action potential on the muscle contraction, whereas a cholinergic synapse could be inhibitory if chloride ions are being released and chloride ions enter. We also have another comparison. We've got the neuromuscular junction is between a motor neuron and a muscle fibre, whereas the cholinergic synapse is between two neurons. The neuromuscular junction is the end point for the action potential, because once it hits that muscle, the muscle contracts and then it doesn't move, the action potential doesn't move any further. Whereas with our cholinergic synapse, the action potential is generated in the next neuron, and then that could be carried on to another neuron afterwards as well. And lastly, with the neuromuscular junction, it's that point we were just saying previously, knowing exactly where the neurotransmitter binds. So on a neuromuscular junction, it binds to the sarcolemma, or the receptors on the sarcolemma, I should say, and you could also phrase it as receptor on the muscle fibre membranes, because that's what a sarcolemma is. Whereas in the cholinergic synapse, it binds to receptors on the postsynaptic membrane of the neuron. So some examiner's tips then linked to specifically neuromuscular junction questions that have come up previously. And I actually have looked through some of the feedback from examiners, and there was one question recently, which was a 4-mark question about what happens at a neuromuscular junction. And of those 4 marks, only 7% of students got those 4 marks. So they got 4 out of 4. 3/4 approximately did manage to get at least 1 mark or higher. But what this shows us is these types of questions students generally find pretty hard to get high marks on. So if you can learn this theory really well, go through all those key marking points we said, and look at these common mistakes to avoid, then that should place you in those top percentage of students that are able to get the full marks. And therefore on these discriminated questions, which means discriminating between the high and the lower marks that students get, you're more likely to get more marks across the whole paper if you can get higher marks on these types of questions and therefore improve your grade. So common mistakes to avoid. First of all is read questions like this really carefully, because going through what happens at a neuromuscular junction is a long process. So often they split it into what happens after the neurotransmitter has been released, or what happens in the lead up to the neurotransmitter being released. So make sure you read the question carefully so you're only answering about exactly what they've requested, so you don't end up wasting time writing about everything. Next then, students often write the neurotransmitter moves across. That won't get you a mark, because instead of saying move, you have to say it diffuses across the neuromuscular junction. That's the same with that as synapse as well. The next point is that students, when talking about what the neurotransmitter binds to once it's diffused across, is incorrectly saying it binds to the muscle, rather than specifically saying it binds to receptors which are on the sarcolemma. So you would have to say it binds to receptors and that those are located on the sarcolemma. Next point we've got is that students often correctly mention that the sodium ion channels open in the sarcolemma, but then they forget to say that that means sodium ions can diffuse into the muscle. So making sure you do so the sodium ions diffuse in. And then lastly is 1 mark that often gets completely missed off right at the end of the process is knowing that that release of calcium ions can then occur from the sarcoplasmic reticulum, which then leads to the sliding filament theory. And that takes us on to the next bit, which is looking at the ultrastructure of the myofibril, which is where the sliding filament theory occurs. So you need to know these key structures, so you could turn this into flashcards if you wanted, so you've got those key terms, but let's just go through some of these structures and we can have a look at it on the diagram. So the muscle is composed of muscle fibres, and these are long cylindrical cells containing lots of nuclei. And within these fibres, these are the key structures that are all involved in muscle contraction. So we can see here we've got myofibrils, and these are long cylindrical structures within the muscle fibres, and they're made up of repeating contractile units called sarcomeres. So lots of sarcomeres are joined together to make a myofibril. Lots of myofibrils are joined together to make a muscle fibre. And the myofibril, which contains the sarcomeres, contain the proteins actin and myosin. And it's the actin that slides across the myosin during a muscle contraction. Then we've got the sarcomere, which we can see down here. We've got one sarcomere shown. This is that functional unit of a myofibril, which is defined as the region between two Z lines. So we've got the Z line here, Z line there, that indicates one sarcomere. And within your sarcomere, you have actin overlapping the myosin proteins. And actin is the thin filament, myosin is the thick filament. The sarcolemma, we've got pointed out here, that is the membrane of the muscle fibre, which we were just talking about because we said that is where you have the receptors that the neurotransmitter binds to at a neuromuscular junction. And when you have that binding, that is where we have the conduction of the action potentials to help trigger the contraction of the muscle. Next then we have sarcoplasmic reticulum, which we've zoomed in here and we can now see this sarcoplasmic reticulum. It's a specialised form of smooth endoplasmic reticulum that surrounds the myofibril. It can store and then release calcium ions, which are essential for the triggering of the muscle contraction in the sliding filament theory. And then lastly, the sarcoplasm. This is the cytoplasm of a muscle fibre, basically. And it contains organelles, enzymes, myoglobin, needed for energy release and muscle contraction. And it's also where you have glycogen and ATP synthesis. And we're going to be looking at the role of those shortly. So here is our sarcomere. That's the same information that we just had on the previous slide, but it's just showing you in a bigger image. Here's our sarcomere. We've got our Z line, and then we've got the other Z line to show where we have the sarcomere boundary. And we're going to come back to this image here where we've got this diagrammatic version showing H zone, I band. I'm going to come to that a bit later in this video because you do get questions where you have to say what is meant by an H zone, an I band, and what happens to the thickness or the distance of those when muscles contract. We'll come back to that shortly after we've gone through the sliding filament theory. So this is how the muscle contracts. So when an action potential reaches a muscle fibre, it triggers the release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm, which we briefly touched on at the very end of what happens at a neuromuscular junction. Those calcium ions bind to tropomyosin, which is shown here in orange, which is a protein which normally blocks these myosin binding sites on the actin filaments. But this binding of the calcium ions causes the tropomyosin to move, and it exposes those binding sites on actin. Because that's now exposed, we get this cross-bridge formation. And this is when our myosin molecule, which has these myosin arms and the heads, it has ADP and PI attached to it. And the myosin head is complementary in shape to that binding site, so it can bind to those exposed binding sites on the actin. And when it is bound, we call this an actinomyosin cross-bridge. That is a key marking term, that's why it's highlighted. Then that takes us on to the next step, which is the power stroke, which is when the actin filament is moved. So once that cross bridge is formed, we can see that the myosin head has bent or it's pivoted. And that is when it's pulling the actin filament along. And this is known as, or this movement is known as the power stroke. And when that power stroke happens, it releases, or it causes the release of the ADP and PI. And it's generating tension, and that is what's causing that actin filament to slide over the myosin. Next then, we have the ATP binding and the myosin head detaches. So at this point, a new ATP molecule binds to the myosin head. That causes a slight change in shape, so it detaches from the binding site on the actin filament. And this ensures that the myosin head doesn't remain permanently attached, and therefore it can reattach and slide the actin even further, causing more muscle contraction. So the next step is ATP hydrolysis, and the myosin head resets back to its original position. Within the sarcoplasm, which we said is like the cytoplasm, the enzyme ATPase is present, and that enzyme is activated by the presence of the calcium ions. ATPase hydrolyzes ATP into ADP and PI, and that releases energy. That energy released from that ATP hydrolysis is what causes the myosin head to reposition or recock back to its original state, and that would be able to form a new crossbridge. So this cycle continues as long as you still have the action potential. Therefore, the calcium ions are still being released and remain high, and that means the tropomyosin is still exposing the binding sites on the actin. So that will happen continuously, pulling the actin filaments towards the centre of the sarcomere, shortening the muscle fibre and causing muscle contraction. When the nervous stimulation does stop, though, calcium ions are then actively transported back into the sarcoplasmic reticulum, and that allows the tropomyosin to move back to its original position, blocking the binding sites and preventing any further contraction. So let's go through some common exam questions or themes that come up in exam questions linked to this topic. One of which is you could be asked to describe the role of ATP or phosphocreatine. So here are your key roles of ATP in the muscle contraction we've just gone through. So number one, it provides the energy to break the actin and myosin bridges, or in other words, the myosin can detach from the actin. It also causes the movement of the myosin head, or that power stroke, so the actin filaments are moved inwards. And also it provides energy for the active transport of calcium ions back into the sarcoplasmic reticulum when the nerve stimulation stops. So if ATP levels are low, that means these processes can't happen, and therefore the muscle can't contract efficiently. And that's where phosphocreatine comes in. This isn't an abbreviation that you tend to see, but I've put it there just in case you do come across it. But in the exam, you would have to say phosphocreatine, do not abbreviate it. Now, this acts as a rapid ATP source because the phosphocreatine can donate a phosphate group to the ADP. So you then have ADP combining with that inorganic phosphate group to make ATP, and that then provides the ATP for all of these processes so that you can then still have these short bursts of intense activity. Another common theme in these questions is what is the role of glycogen granules in skeletal muscle? So for this one, you'd need to say that it's a store of glucose and that it can be hydrolyzed back into glucose. That glucose is important because it can be used in respiration or as a respiratory substrate to provide the ATP that is needed, and that then links back to all of these roles that we just talked there about what ATP is used for. Another common theme is what is the role of calcium ions in the contraction of the myofibril? So specifically what the calcium ions are doing is when that action potential reaches the muscle fibre, calcium ions are released from that sarcoplasmic reticulum and they diffuse into the myofibrils. The calcium ions bind to the tropomyosin. That causes the tropomyosin to move and expose the binding sites on the actin filaments. With those sites now accessible, the myosin heads can attach to the actin, forming the actin-myosin cross bridges, and that's then leading into the rest of the sliding filament theory. So that takes us on to looking at this sarcomere in terms of the bands and the zones. So this is our sarcomere. We've got our actin filament and our myosin filament is shown in blue here, and those lines coming out are the heads. So the A band is the distance of the myosin. The Z lines are the start and end point of one sarcomere. The I band is where you just have actin and it is not overlapping with the myosin at that point. We have our H zone as well. The H zone is where you just have myosin with no actin overlapping it. So those are the key structures that you need to know. And when a muscle contracts, the actin is sliding over the myosin. So therefore, the two Z lines are moving closer together. And that's one thing that we can see happening. Here's the position of the Z line. When the muscle contracts, the Z line has moved inwards. So the distance between Z lines decreases when the muscle contracts. Another thing that we can see happening is the H zone distance also decreases. So the H zone is where you just have myosin by itself and no actin overlapping it. That becomes much narrower or a shorter distance because the actin is sliding inwards. So when a muscle contracts, the Z lines come closer together, the H zones become shorter, and also the I band will become shorter as well, because the I band is where you just have actin by itself, and we can see here that that is shortened. So Z lines get closer together, H zone and I band shorten. The A band is always constant. That doesn't change because that is the distance from one end of the myosin to the other. And the myosin is not sliding, so the A band remains constant. The last little thing you need to know with the muscles is about two different types of muscle fibres. We have slow and fast twitch fibres, and you need to know about the structure, location, general properties, the type of respiration, and a little bit about how that links to their uses as well. So a slow twitch fibre, these are the muscle fibres that are contracting more slowly. They can continue to contract for longer periods of time, and therefore they are useful in endurance activities like marathons. And they contain a large store of a protein called myoglobin, which can store oxygen. They have a rich blood supply to continually provide oxygen, and they have many mitochondria. So basically they are set up for aerobic respiration because it provides lots of oxygen. So that's the primary respiration type, aerobic respiration. So that means that they can generate more ATP because aerobic respiration produces more ATP than anaerobic, but it produces that ATP more slowly. So those are our key properties. It does also have a store of glycogen, but it's a smaller store compared to the fast twitch fibres. And the fast twitch fibres are the muscle fibres that can contract faster. That means that they can produce these short bursts of powerful contractions for intense activities like sprinting or weightlifting. And they can do this because they produce ATP more quickly, but they are going to generate less ATP because it's anaerobic respiration. So they have thicker or more myosin filaments. They have a large store of glycogen. They have a big store of phosphocreatine as well to help make ATP from ADP. They have a high concentration of enzymes as well needed for anaerobic respiration. And an example would be in biceps, and that's linked to weightlifting. We've already said they produce ATP more quickly and they generate less ATP, and that's linked to the fact that it's anaerobic respiration that they primarily use. And they have a large store of glycogen. Both muscle fibres do have stores of glycogen, and that's because glycogen can be hydrolyzed into glucose. Glucose is a respiratory substrate, and therefore you can use that to produce ATP. So a few examiners' tips linked to these fast and slow twitch muscle fibres is, if you do have an exam question about them, make sure you are comparing both muscle fibres and not just talking about one of them. Just say that slow twitch muscle fibres produce a lot of ATP. You'd need to say whereas fast twitch don't produce as much. So make sure it's always a comparison. Next thing that examiners have picked up is that sometimes students are quite vague with their terminology and sometimes even incorrect. So sometimes they've said that fast twitch muscle fibres work anaerobically instead of respire anaerobically. Then there's been the misuse of terms like saying glycolysis instead of glycogenolysis, and glycogenolysis is this process here, the hydrolysis of glycogen into glucose. Then we've got sometimes students missed off this mark altogether, not explicitly comparing ATP production. So they forgot to state that anaerobic respiration produces 2 ATP molecules without mentioning this being less than the amount produced in ATP. Now, you probably wouldn't have to literally state the quantities, but you do need to know that anaerobic respiration produces much less ATP because it only involves glycolysis, whereas aerobic respiration produces far more ATP. And then the last thing is incorrectly saying that energy is produced instead of saying ATP is produced. So energy cannot be created or produced, it can be released. You can create ATP though, because that is a chemical molecule, it's a biological molecule you learn. So that is it for this topic. If you have found it helpful, just to make you aware that my A-level biology notes cover all of the theory, key terms, and my flashcards also contain the key marking points and key terms for the theory. So if you want to have all of that in one place, the links are in the descriptions. But that is it for this video. I hope you found it helpful, and I'll see you in one of the next videos coming up.